Hi class, this is Miss Sheely here to give you your Chapter 3, Section 4 Inheritance Notes. Alright, first to go over some vocabulary that's really important. We of course have our genotype and phenotype. Our genotype is the combination of the alleles of a gene carried by an organism, so it's what you can't see inside of the DNA. But your phenotype is the expression of the alleles of a gene carried by an organism. It's your phenotype, so what you see. Here we have two chromosomes. Our location of the genes on the chromosome is called a gene loci. And in the middle of our chromosome, we have our centromeres, which will hold our two sister chromatids together when those genes are duplicated during cell division. But we have some different types of vocabulary to review that has to do with the alleles. The first most common is our homozygous dominant. This is having two copies of the same dominant allele, typically shown by a big letter, big letter. The opposite of that, homozygous recessive, is having two copies of the same recessive allele, little letter, little letter. Recessive alleles are only expressed in the phenotype when they are homozygous, when you have two of the same alleles. You also have heterozygous, and that is where you have one big letter and one little letter, or having two different alleles, and in that case, the dominant allele is expressed. These alleles are just different versions of a gene. But we also have a new one. We have a codominant. These are pairs of alleles in which both of the alleles are expressed or when they are present. So if you remember back to baby bio, we have the checkered chicken, where you saw both the black and the white. A heterozygous person can also be a carrier of a disease when it's a recessive disease-causing allele. Excuse me. Morpheus would like to have some love. Say hi, Morpheus. Hi. All right. So, Mendel was one of the first scientists to really look at genetics and inheritance, and he just took some yellow pea plants and he crossed them with some other yellow pea plants. And most of the offspring of these two yellow pea plants were yellow. But there were some that were green, and that led Mendel to come up with some ideas. And one of those ideas was an idea of segregation. So, Mendel, in his experiments, he figured out that there was something, heritable factors, what he called them, that were passed on, and that these heritable factors could have different versions. We know now that these heritable factors are called genes, and that these different traits or different versions of the genes are called alleles. But Mendel didn't know any of this. But he was able to figure out that there was something different about these yellow pea plants. We know now that they must be heterozygous because we see the yellow phenotype, but we get this recessive green allele. So through meiosis and fertilization, some of our offspring are homozygous recessive, or the green color. So this idea of segregation is that the alleles of each gene separate into different gametes when the individual produces gametes. Well, let's explore this idea of segregation a little bit further. All right, so here we have our yellow pea plant again. This time one is indicated as a female and one as a male. And their genotypes, as we've already talked about, are heterozygous. So if the alleles segregate or separate into different haploid gametes, then we would put them here on our Punnett square. So we have our male, big Y, going here in the first spot, and our little y of our heterozygous combination here, and then our female going off to the side. So fertilization is when we take these haploid gametes, so each of these on the outside of our Punnett square is where the egg or the sperm would be. So those are haploid cells. They only carry one of the chromosomes. But fertilization is when we get a diploid zygote. So a Punnett square can be used to show the possibilities from fertilization. And when we get from this fertilization, when we fill out our Punnett square, we get these results. And how do these results break down? Well, we have 3 of the yellows and 1 of the recessive green. But as you can see, we have 4 different combinations of the genotypes. We typically write the ratios in the simplest mathematical form, and we usually focus on the ratio of the phenotype because that's what we can see. So in this case, we have a 3-to-1 ratio. All right, we can also, and this is called a monohybrid cross because we're looking at a single trait. So this is just yellow versus green. There are more complex crosses, such as dihybrid crosses. You might remember those from your baby bio classes. All right, sometimes we don't know what one of the genotypes are, and we can use a process called a test cross to determine the genotype of an unknown individual. The unknown is crossed with a known homozygous recessive. So what does that mean? Well, we have a red flower, and we know that red is dominant, but we don't know if this is big R, or big R, little r. We know that one of them has to be a big R, though. And we're going to cross that red flower with a white flower. And the white flower has little r, little r. So when we cross these two, there are 2 possibilities, 2 Punnett square outcomes. We could get this possibility if our red flower is homozygous dominant, we would get all red phenotypes. But if our red flower is heterozygous for red, meaning it has a big R and a little r, then we would get some red, or approximately 50% red, and some white, approximately 50%. So if we breed these 2 flowers together and we get any white, then we know that our red flower must be heterozygous. If we get all red, then we can say that the parent is probably homozygous dominant, but we don't know for sure because it is just a 50-50 shot. So that's what a test cross is, so we can use to figure out an unknown genotype. The next thing we're going to talk about today is called a pedigree chart. So pedigrees can be used to trace family histories, and we can deduce genotypes or the risk of an inherited gene-related disorder. So a pedigree is basically a family history, but looking at the genes. So there's a couple of things you need to know. First of all, squares are boys and circles are females. Horizontal lines across mean that they were married or had children. Not necessarily married, but that they had children together. And the vertical line indicates the difference between the generations and the children. So parents A and B had children C and D. Both of these children were married or had children of their own. So C had 1 child and D had 3 children. Then you need to be aware of the key. Most pedigree charts will show the affected with a solid shading and a non-affected with a white or non-shaded. And then deceased does not mean diseased. Deceased means the person has actually died. Doesn't have anything to do with the genotype, but if you're looking at a disorder or a disease, that could be some interesting information. All right, so let's try and deduce the genotypes for A and B. Well, you see that they're not affected, but do we know if they're heterozygous or homozygous dominant? Well, they both had 2 children. Okay, so A and B had children C and D as previously established. Well, child C is affected with no enzyme, which is the key here. So we would say that A and B are both big T, little t. The trait is recessive, and since both of the parents are normal, they have produced a child with the affected issue, which is having no enzyme. So they have to be big T, little t, so that they can both give a little t. All right, Now let's figure out what C is. Well, C is affected, and this is a recessive trait, so it is going to be, C is going to be little t, little t. Recessive traits are expressed only when they are in a homozygous combination. Well, let's figure out what D is. D came from both of the parents, okay? And D, but it had an affected child. So just like A and B, D has to be heterozygous as well. Otherwise, child H wouldn't have been affected, okay? All right. So here's an example using an actual disease. This is a clinical example. So you can use pedigree charts to trace family histories like we just said. This is a pedigree for a disease called Phenylketonuria or PKU. So you have two sets of families in this case. So parents A and parents B, and then their parents with them as well. Excuse me. So take a second and see if you can figure out what's going on here. Is PKU a dominant or recessive disorder? And how do you know? So again, this one is just like the other, where the affected is shaded and the non-affected is not shaded. And the disease, so we have 3 of the grandparents that are deceased. All right, so now that you've had time to think about that, all right, so looking at it, when you're looking, you have a child, child A from generation 2, has the affected disorder and came from a parent that was not affected and a parent that was affected. So that is some indication that it is recessive. The same with over here. You have an affected parent with a non-affective parent having a non-affected child. So this is a recessive disorder. The unaffected mothers in generation 1 had an affected child here, so the mother here must have been a carrier. All right, so what is phenylketonuria or PKU? This is a missense mutation in the gene that produces tyrosine hydroxylase, and this enzyme converts phenylalanine to tyrosine. So the phenylalanine builds up in the body. The problem with the phenylalanine buildup in the body is that it results in developmental problems and seizures. Excuse me. It is a progressive disease, so you must find it and treat it early. Dairy, breast milk, meat, nuts, and aspartame must be avoided as they are rich in phenylalanine. This is how we test for phenylalanine or phenylketonuria, PKU. We do a foot stick at birth, and this would be the card with the blood samples. Here is a video clip. This is a 6-minute video clip from YouTube with a boy with PKU. I'm not going to show this now, but it is linked to your notes in Blackboard. So check that out if you're interested. All right, some more information. So let's review some genetics. What is a missense mutation? All right, so we have a base substitution mutation where the change in a single base results in a different amino acid being produced in the polypeptide. It is an autosomal disorder, chromosome 12, and the locus of the tyrosine hydroxylase gene is chromosome 12, lower arm between the 22 to 24 area. All right, sometimes, like I said in our vocabulary at the beginning, we have genes with 2 or more alleles, and sometimes they are codominant, where both alleles can be equally expressed. And human blood typing, ABO blood typing, is an example of multiple alleles and codominance together. So this is an example that you need to be familiar with. The gene for the blood type is actually for the cell surface antigens or the immunoglobulin receptors. These are either absent or present. If they're absent, then it's type O. If they're present, then it's A B, or AB, as you can see in the flow chart here. No antigens, type O. Yes, antigens, you can have an A type, a B type, or both A and B. When the genotype is heterozygous, when it's A and B, then this is codominance because you have both the A antigens and the B antigens. Your alleles, if you remember, are little I for no antigens present, big I with a superscript A for type A antigens present, and big I with a superscript B for type B antigens present. So antibodies or immunoglobulins that sit on the surface of the red blood cells are specific to the antigens. The immune system recognizes foreign antigens and produces antibodies in response. So if you're given the wrong blood type, your body might react fatally as the antibodies will join of your blood, will join with the blood that's given to you, causing it to clot. Because of that, blood type O is known as a universal donor because it has no antigens which the recipient immune system can react with. Type AB is a universal recipient as it has no antibodies which will react to AB antigens. Okay, so here you can see here, if you have the square shape, in this case, for type A on the blood cell, then you have the opposite antibodies in the blood. If you had the B type with the pointy antigens, then you have the square-shaped antibodies. If you have both antigens on your blood cell, then you're not going to have any antibodies, which is why AB is a universal recipient. Those antibodies, there's no antibodies to react to any of the blood. And if you have type O blood with no antigens, then you have both the A and the B plasma antibodies. When they give blood, blood type O is a universal donor because it has no antigens on the red blood cell, and the antibodies are centrifuged off and separated. So you just get plasma blood cells when you give blood. All right, so changing gears just a little bit. So we talked about codominance right there. Let's talk about sex-linked genes. So some chromosomes are autosomes. Those would be your 1 through 22. Your 23rd set in humans are your sex chromosomes. And remember that they are non-homologous. There are many genes on the X chromosome because it is bigger. There are less genes on the Y chromosome, but the genes that are on the X chromosome aren't present on the Y chromosome because of the size. So sex-linked traits are those which are carried on the X chromosome in the non-homologous region, so the same region, not the same. So you have both an upper and lower part of the X chromosome. You can see here the non-homologous region that is not the same. Some common examples that IB wants you to know about of sex-linked genetic disorders, you have hemophilia and color blindness. We're going to talk about color blindness first. So, looking at these two numbers, well, they're both the same. Looking at this this picture, what numbers do you see? You're either going to see a 5 or a 2. If you see a 5, then you have normal vision. If you see a 2, then you are red-green colorblind. The red-green gene is carried at locus X for the X chromosome, lower arm Q, and at spot 28. This locus is in the non-homologous region, as you can see here, so there's no corresponding gene or allele on the Y chromosome. Normal vision is dominant over color blindness. So if you have a normal female, we usually indicate this with a capital N. So a normal female would have 2 X's with a capital N superscript. Excuse me. An affected female, a colorblind female, would have to have both the X's to have the lowercase n in the superscript. But you could be a female that is a carrier, which means that she carries the gene for color blindness, but only on one of her chromosomes. In boys, because males only have one X chromosome, then they are either normal or affected, because the Y chromosome carries no allele for this gene, and we do not therefore write an allele. So females can be homozygous or heterozygous with respect, and then heterozygous females are called carriers. All right, so how do we write this? Let's look at a chance of a colorblind child in the cross between a normal male and a carrier mother. So we would first segregate our genotypes and then fill in our Punnett square. So our results are that we would have a normal female, a normal male, a carrier female, and an affected male. So there's only a 1 in 4 chance to have an affected colorblind child. What ratios could you expect to see in a cross between a colorblind male and a homozygous normal female? Or what about a normal male and a colorblind female? You need to be comfortable with doing these types of Punnett squares. A little bit more on red-green color blindness. There are 2 genes, and they're both found at the same location. They are responsible for producing photoreceptive pigments in the cone cells in the eye. If one of these genes is a mutant, then the pigments are not produced properly, and the eye can't distinguish between green and red wavelengths in the visible spectrum. Because the gene is in the non-homologous region when compared to the Y chromosome, the red-green color blindness is known as a sex-linked disorder, because the male has no allele on the Y chromosome to combat a recessive faulty allele on the X. So red-green color blindness in men is caused strictly by the mothers. But red-green color blindness in females has to be caused by both parents. All right, let's look at the other sex-linked disorder. This is hemophilia. Blood clotting is an example of a metabolic pathway. It's a series of enzyme-controlled biochemical reactions, and it requires globular proteins called clotting factors. A recessive X-linked mutation in hemophiliacs results in one of these factors not being produced. Therefore, the clotting response to injury does not work, and the patient can bleed to death, even from a bruise. So just like in the color blindness, we're going to use capital letter H for the healthy clotting factor and lowercase h for no clotting factor, thus leading to hemophilia. So you can again have a normal female with 2 capital H's, an affected female with 2 lowercase h's, or a carrier female that is heterozygous. And again, males cannot be carriers. They are either normal or affected. A little bit more about hemophilia. So this is your pathway. You have your damage. Your clotting factors start. They are the ones that take a protein called prothrombin. They turn it to thrombin. And then thrombin turns something called fibrinogen into fibrin. And fibrin is what causes the clot. So this is what you want. But if you have hemophilia, this whole pathway doesn't happen. So there are treatments for hemophilia, and that is to actually inject clotting factors, these clotting factors here, to start the process. So we do this, we make this in the industry. We find a gene for healthy human clotting factor, and we cut it out. We insert the gene into sheep milk gene in sheep embryos. Then we extract the clotting factor from sheep milk, and we purify and produce the injections for patients to get. But we didn't know that a long time ago. We didn't have the science and the research in order to be able to do that. This pedigree chart is of the English royal family, and it shows the inheritance of this X-linked disorder called hemophilia. So you have normal males and females being unshaded, carriers being half shaded in this case, and then hemophilias to be fully shaded. So you can see here that the fully shaded were just in the men, and the women were all just carriers. All right, so that's it for today's inheritance notes. Thanks for watching, and look forward to the next ones.